So I want to welcome you all to this 11th session of our summer school, The Virtual Brain in Clinical Research. My name is Christoph Hüttl and I'm an MD PhD student at Charité and the Berlin Institute of Health in the Brain Simulation section led by Professor Petra Ritter. And it's my great pleasure to do this session together with one of my PhD supervisors, Dr. Konstantin Bülau, who is a postdoc in our lab and also a resident in the Department of Neurology at Charité. Our talk is titled Modeling Mechanisms of Psychosis. And before we get to dive right into the exciting science, some organizational reminders. As you know, this summer school is being awarded with two, respectively 3 CTS points, depending on your program. For this, you will have to complete a 40-question multiple choice exam. There will only be one correct answer per question. And I think if you have attended all the sessions or listened to them afterwards, you will be definitely feasible to answer these questions. The exam will be distributed via email after the last session, and then the results may be returned via the provided answer sheet via email to our PI, Professor Ritter. You will get her email address then also in that email. And then I think most of you have also already attended the great talk by Dr. Leon Stefanowski on integrating biological knowledge in brain network models and modeling neurodegeneration. And we are going to build on some of the tools mentioned in these talks, especially the virtual brain adapter of semantics. And we are going to give some use cases, some additional use cases for these great tools. So if you have seen this talk already, that's wonderful. We are going to build on this. And if you didn't have the chance to take a look at this yet, you will dive right in with the interesting use cases. And then maybe after our session today, you can freshen up some of the basics and the underlying workings of these great tools and overall the great talk by Leon. So today, this talk is going to be all about psychosis, and we are going to talk about the definition of a psychosis, of the causes, about the developmental factors, and about the underlying mechanisms, including immunology. But first, I actually want to talk about something else, because when reading some of the neuroscientific literature with regard to psychosis, one thing is not always so clear, and that is the differentiation between symptoms, syndromes, and disease entities. And for this, I want to give you a practical example. In Germany, every newborn baby receives a screening test after they are born, testing for a series of both severe and treatable genetic diseases. And one of those diseases is called phenylketonuria. And this is a disease where, due to a genetic abnormality, an enzyme is losing its function. This enzyme is called phenylalanine hydroxylase, for short PAH. And usually this enzyme catalyzes the reaction from phenylalanine, which is an essential amino acid, to tyrosine. And as this enzyme is not working properly in this condition, phenylalanine cannot be transferred to tyrosine and is accumulating. And then it's broken down via various steps to, among other things, phenylacetate. And this is a substance which is toxic to the brain. It leads to cognitive deficits with regard to learning, memory, concentration, problem solving, and all these different kinds of symptoms which arise from this disease. And in this case, the treatment consists of a special diet, so a reduction in the amount of phenylalanine that is being taken up through the diet. So in this case, we have a very clear understanding and association about both the pathophysiology of the disease, the mechanisms, the symptoms, and then the therapy. And this is possible because this is a so-called monogenetic disease. So there's one rather clear genetic cause of this disease. But unfortunately, not all types of disease are monogenetic or even monofactorial. Most diseases, including most mental disorders, are so-called multifactorial diseases, which means that there are multiple factors being relevant for this disease, including, of course, genetic factors, but also proteins that may be misfolding, environmental and societal factors are relevant, epigenetics play a role, inflammation plays a role, signaling changes play a relevant role. So multiple factors coming together in these multifactorial diseases, leading to a clinical phenotype. And to give you an overview, mental disorders are defined as of DSM-5 as a syndrome characterized by different clinical features, including significant disturbance with regard to cognition, emotion regulation, or behavior. And what is also important is that they significantly impair the quality of life, the abilities with regard to daily living. And now we have already heard an example for different symptoms, including memory deficits, for example. And these symptoms can then be grouped together to syndromes, being characterized by a number of symptoms that often occur together, are somewhat related mechanistically or otherwise bundled together. And one relevant syndrome that we are going to talk about today is the psychotic syndrome. And the psychotic syndrome consists of three symptom categories. The first one are the so-called positive symptoms. And they are not called positive symptoms because they are somewhat pleasurable for the patients, but rather because they add something upon the usual perception and way of life of healthy individuals. In this case, this is mainly with regard to hallucinations or delusions. Then there are also the so-called negative symptoms, which take something away. So they are characterized, for example, by affective flattening, by social withdrawal, by apathy, and also reduced speech production, also known as alogia. And then as the third category, they are the cognitive symptoms with deficits in learning, memory, problem solving, and so on. And together, these symptoms of psychosis form the so-called psychotic syndrome. And these symptoms or clinical features that are bundled together and under this label, under the syndrome, of course, occur as part of the psychotic syndrome, but they are not specific to one disease entity. So what most people maybe think about first as a disease entity with regard to psychosis, maybe schizophrenia, and this is definitely very important, and we're going to talk about this in more detail very soon, but this is only one of the disease entities that may be relevant here. Substance use disorders can be relevant, childhood developmental disorders, mood disorders, and anxiety disorders may also fulfill many of these symptom categories. And to give you an overview, we can take a look at this list of many of the disease entity which can lead to a psychotic syndrome here. So the first one is schizophrenia, as we've already talked about, but also bipolar disorder with psychotic features may be mentioned here. Major depressive disorder with psychotic features is also relevant here, especially with regard to the negative symptoms. There's a relevant overlap and also a big comorbidity of up to 50%. Shown in the literature. But then there are also other types of diseases, including psychosis induced by substance abuse, for example, but then also, and this is also important, psychosis due to medical condition, including neurologic conditions. And one example for these neurological conditions is the so-called autoimmune encephalitis. And one example for autoimmune encephalitis, which is also the research here is being led here at Charité, is the so-called anti-NMDA receptor encephalitis. And here we have, I think, a really beautiful picture where we can see already the root of the problem, because here we can see that the anti-NMDA receptor antibodies in green are significantly increased on a hippocampal neuron, which is colored in red here, and the nucleus in blue. And in this condition, we have a rather clear understanding, as far as we can, of course, assess it, about the pathophysiology of this disease, because we have these autoimmune antibodies against the GLUN1 subunit of the NMDA receptor. And these autoantibodies travel into the brain and are present in the brain, and they can bind to this NMDA receptors, which then leads to an internalization of the receptors, which results in a reduced receptor density and reduced glutamatergic signaling on the relevant synapses. And these types of autoimmune encephalitis are often associated also with oncological diseases, including for NMDA receptor encephalitis, especially the teratoma of the ovary. And one, at least in Berlin, a very well-known figure is depicted here, the ice bear. His name was Knut, and he was a big star here in Berlin at the time in the Berlin Zoo, and he was really beloved by the Berliners. But unfortunately, he died at a rather young age, and through abduction, it was found out that this was actually due to NMDA receptor encephalitis. And you could actually observe some of the symptoms that we also see in humans in his behavior at the time. So maybe that's a way you can remember this. So first we have a prodromal phase of around 1 week where symptoms are not so severe yet, but then really the intense psychotic symptoms develop, including delusions, hallucinations, also mania, disorganized behavior, thinking, insomnia, and so on. And then even a coma state can occur with even more severe also neurological complications, which is kind of the peak of the symptoms. And then later throughout time, you reach more or less a plateau phase with prolonged deficits with regard to executive function, emotion regulation, increased impulsivity, sleep abnormalities, and so on. And then coming back to the human medicine side of things, the diagnostic criteria, of course, there are plenty. Seizures also can play a relevant role here, but the only definite diagnostic criteria are IgG antibodies in the cerebrospinal fluid. But of course, with regard to autoimmune encephalitis, NMDA receptor, the NMDA receptor is not the only relevant target. There's also the LGI1 encephalitis or the Casper2 encephalitis, just as two examples. So here we have this very rather clear immunological disease pathway with the relevant autoantibodies. But of course, this is relevant in some forms of psychosis, which then also enables us to give specialized treatments. But of course, there are also other disease entities that can cause psychosis. And one of the big ones is schizophrenia, which we are going to talk about now. So schizophrenia shows abnormalities with regard to one or more of these domains. We have already talked about this with regard to the symptom categories. This includes delusions, hallucinations, disorganized thinking, disorganized motor behavior, and also the negative symptoms. And this can, of course, as we mentioned, have many different results. But in this case, schizophrenia is defined as the so-called idiopathic psychosis. And idiopathic is coming from the Greek idios, one's own, and pathos, suffering. And together, this means that an idiopathic disease can be any disease with an unknown cause that seems to appear spontaneously. So we don't have a really clear understanding of the origin of the disease. And then the diagnostic criteria of schizophrenia include the different symptoms that need to be present, at least one of the main symptoms, including delusions, hallucinations, and disorganized speech. Then also the qualification criterion has to be met, which means that it has to significantly impair the levels of functioning, the daily living. Then, of course, the time course also has to be at least 6 months, at least one of them with the most severe symptoms, or less if it has already been successfully treated before. And then, as we said, schizophrenia is defined as the idiopathic psychosis. So we need to rule out other differential diagnoses, such as the NMDA receptor encephalitis, for example, because then we have a clear understanding of the underlying mechanisms, and we have to rule this out, of course. So taken together, schizophrenia is a clinical diagnosis. Schizophrenia is also a disease entity with a really relevant prevalence of around 1% lifetime prevalence in the worldwide. To give you a more comprehensible number, at Charité, we have around 300 medical students per semester. This means around 3 students from each semester will develop schizophrenia throughout their lifetime. And at Charité in general, we have over 20,000 employees. And that means, just from a statistical point of view, that 50 of the employees at Charité are suffering from schizophrenia, and every year one new case will appear. So this really is a relevant number of patients throughout the whole world. And the lifetime prevalence is about equal in men and women, and while men show more severe disease trajectories most of the time, also having an onset of the disease that is slightly earlier. On average, usually the first episode of psychosis occurs in the early 20s. And the life expectancy in patients suffering from schizophrenia is significantly reduced up to around 15 years. And beyond the personal suffering, which is, of course, the main focus of our medical work, this disease is also associated with relevant societal costs of around 150 billion every year in the US alone. And for this, I also want to give you an overview about the disease development. Here you can see that the relevant risk factors already begin before the birth of the individual, with genetic factors playing a role, but also prenatal and perinatal factors, including infections. Then living in an urban environment and also migration plays a crucial role for the disease development. And then the very crucial time is in the late teens, where additional risk factors can occur, including social isolation, stressors, and also, which I really want to emphasize, drug abuse, especially cannabis, plays a relevant role here. And then the usual time course would be that in the early 20s, when maybe some additional stressors accumulate, and you have this first bloom up, usually of the positive symptoms, which lead to seeking attention by the medical system, so having severe hallucinations and delusions. And usually these positive symptoms can be treated rather well, but then, of course, you will have to find an ideal treatment regime, and you will often experience more than one episode. But after some time, you will find a fitting therapeutic avenue, reaching more or less a plateau phase. But then, of course, crisis and such situations can lead to additional episodes, but the long-term goal would be the relapse or the remission of the disease. And considering the developmental aspects of schizophrenia, we can also observe these objectively measurable changes in the brain. And this includes deficient myelination already starting around the age of 5. And then we also have an excessive excitatory pruning, especially with regard to excitatory synapses in the prefrontal cortex. And then we can also see the reduced interneuron activity of the inhibitory synapses in the prefrontal cortex. And then what is also important for me to stress is that the time course of the different symptom categories is different. So, as I mentioned, the first symptoms that usually occur are with regard to the so-called positive symptoms. So this is this first bloom up where the first hospitalization usually happens. But these can actually be treated rather well throughout the disease condition, throughout the disease progression. And it's more negative and in the long term also crucially the cognitive symptoms that significantly reduce the quality of life of our patients. And this then, of course, is really significant. As I mentioned, there are also multiple immunological aspects that are relevant for the development of schizophrenia. And these already start before birth. And we have found in big studies and meta-analysis that infections of the mother, especially during the first trimester of the pregnancy, for example, with Toxoplasmosis gondii, but also with influenza virus, can significantly increase the development of schizophrenia of the newborn throughout their life. And this is most likely due to some changes in the immunological signaling during the early phase of the pregnancy, where also the early brain development takes place. So you have difference in the different cytokines, the pro-inflammatory signaling molecules of the human body, including TNF alpha, tumor necrosis factor alpha, and then also interleukin 1 beta and interleukin 6, to give some examples. Then we also know that autoimmunity plays a relevant role. We have already talked about this with regard to NMDA receptor encephalitis, but also a family history of autoimmunity has been identified as a relevant risk factor. Then we have different genetic factors, which I will also go into more detail about, but also genetic changes with regard to the cytokine dysregulation play a relevant role. But then not only the cytokines, also the immune cells, especially the T cells themselves, also can be relevant with regard to genetic alterations. And so we have these different stages of the disease, with first the prodromal phase, then the subacute phase, and then the different episodes. And this leads to a so-called window of opportunity, usually ranging from the late teens to the early 20s, where we already have some signs and symptoms of a disease, and those risk factors are already present. But ideally, the full picture of schizophrenia, the full psychotic syndrome, has not yet emerged. And so this is really a crucial window of opportunity, where also the newly founded Einstein Center for Youth Mental Health, which we are also proud to be a part of, focuses their research interest on. And then considering the treatment. So today's therapies mainly focus on managing the cardinal symptoms. As I mentioned, we already have some very powerful strategies combating especially the positive symptoms, but also somewhat the negative symptoms with pharmacological treatment strategies with those antipsychotic drugs. Um, that of course have side effects, but that can be really relieving to the patients, especially when they are suffering from these strong hallucinations and delusions. But especially with regard to the negative and even more the cognitive symptoms, we are still um, performing research to find more effective treatment strategies. Then tomorrow's therapies have the goal to already be present in this window of opportunity that we have just talked about, and already target individuals which have a high risk for developing psychosis in the future, the so-called clinical high risk cohort, which I will go into on the next slide, and young people who may transition into psychosis in the future. And then looking even more into the future, future therapies may even target these early developmental anomalies, these latent factors inherited through genetics or early life stressors. And of course, it would be outstanding if we would have possibilities for disease modification already at such an early stage. And so, as I mentioned, these clinical high risk for psychosis individuals are very relevant with regard to future treatment strategies, and they are defined as meeting at least one of the ultra high risk inclusion criteria, which are defined by brief, limited, intermittent psychosis symptoms, for short BLIPs, and or attenuated psychotic symptoms, APS, the genetic risk and deterioration syndrome, GRD, and the basic symptoms, including cognitive deficits and some of the negative symptoms. And together, this clinical high risk cohort captures the potential of pre-psychotic or the prodromal phase. And then these individuals have a conversion risk of around 10 to 15% per year of developing the full picture of psychosis, which is, of course, highly elevated with regard to the general population. But on the other hand, it's also not high enough to warrant, and there have been many studies on this already, an early treatment based on this clinical high risk stratification. And so there are multiple approaches going on right now to develop a methodology to identify individuals with a high likelihood of transitioning to psychosis. And one of those projects is led by Professor Nikos Koutouleros, and we are very happy to collaborate with him and his great team. And Konstantin is also going into some of the details about that collaboration later during this talk. And to give you an overview of how such a transition may take place, so you have this pre-morbid phase where maybe you have very few symptoms occurring, and then starting in the prodromal phase, you have some of these basic symptoms with regard to maybe strange thought content, maybe already a little bit of social withdrawal and these things, which may not be very apparent during the time, and rather post hoc, looking back after the diagnosis is made, you then identify, oh yeah, well, a couple of months or years back, actually, yes, he or she has changed his or her behavior, and his functional state was in not such a good shape as we knew it before. And then we have the late at risk of psychosis state, where we already have the attenuated positive symptoms, and then also the brief, limited, intermittent psychotic episode, which then often is the last step before we meet the full transition criteria for early psychosis. And then, as I have mentioned before, schizophrenia is a multifactorial disease, so there are many different pathways leading to this disease entity, and we have not understood everything yet. But at the other hand, on the other hand, there are also many things that we do know, and I want to give you an introduction to this now. So one of the oldest hypotheses with regard to the underlying mechanisms of psychosis is the dopamine hypothesis. And this postulates that changes with regard to the dopaminergic transmission lead to the symptoms of psychosis. And to give you a general understanding, dopamine is a highly relevant neurotransmitter in the brain because it gives meaning to stimuli. It gives them this so-called salience. So what does it mean? At any given point in time, there are thousands of stimuli you take up, you receive, and that are processed in the brain. So right now, for example, you probably have the screen in front of you. You can see my talk, you can hear my voice, but at the same time, there are also so many other things going on in your room. Maybe when you look out the window, you can experience so many other things at the same time. But yet, hopefully, you are able to focus on my voice, focus on the talk. And one mechanism that is really important for this is giving salience to specific stimuli through dopamine. And in general, in psychosis, you have an increased dopaminergic transmission, meaning that generally speaking, too many stimuli receive the added information of being relevant with regard to you in this situation. So, whilst we normally maybe would see 3 black cars passing by, once we look at the street and not think much about it, for a patient suffering from schizophrenia, through these increased levels of dopamine, they think, oh, this is a special sign for me. These 3 black cars, they must mean something. Or when they hear something in the radio, the broadcaster speaking about a certain topic, they potentially think, oh, maybe there's a hidden message, especially for me, and I need to decipher it and so on. So this is a general explanation of why this increased dopaminergic transmission is relevant in psychosis, but our understanding now has moved beyond this, differentiating this into 2 different pathways, both starting in the ventral tegmental area. One pathway going to the subcortical and limbic regions, including the striatum and the nucleus accumbens. This is called the mesolimbic dopamine pathway. And here we see an increase of dopamine type 2 receptors, which are inhibitory G protein coupled receptors. And so here we can then perceive an increase in inhibition, which leads to less regulation and processing of emotions and memory, which is then associated with positive symptoms. And on the other hand side, then we have the mesocortical dopamine pathway linking the ventral tegmental area with the areas of the cortex, especially in the prefrontal cortex. And here we have actually a decrease of another type of dopamine receptors, the dopamine D1 receptors, which has a stimulating G protein coupled receptor. And this decreased stimulation then leads to less cortical planning and is associated with the negative and cognitive symptoms of psychosis. But of course, since the dopamine hypothesis has been published and the research here is going on, but we have also found out that dopamine is not the only relevant neurotransmitter for this disease, and also glutamate and GABA play a relevant role here. And this also makes sense taken together, because for dopamine, we know that some of the most valuable treatments, especially as I mentioned with regard to the positive symptoms, are the dopamine antagonists, those antipsychotic drugs are usually having an anti-dopaminergic component. Then we also know that for anti-NMDA receptor encephalitis, these autoantibodies target those receptors which are relevant for glutamatergic transmission. And then we also know that there are GABAergic changes leading to an altered excitation inhibition balance. And as I briefly mentioned before, genetic factors are also highly relevant with regard to idiopathic psychosis or schizophrenia. It has a high heritability of around 50% concordance rate between identical twins, meaning that if you have an identical twin and they have developed schizophrenia, you have a 50% chance of also developing schizophrenia throughout their life, so massively increased risk. Incidences are 10 to 15 times higher for people who have parents or siblings with the psychotic disorders. And there's also an increasing overlap with regard to evidence. There's increasing evidence for an overlap with regard to the genetic susceptibility in patients both with schizophrenia and bipolar disorders. And of course, there have been many big studies, and research is also still ongoing here with regard to polygenic risk scores. And now we have identified over 100 genetic risk factors through these big GWAS studies. And we know that many of these genes that are relevant here are involved in glutamatergic and also dopaminergic transmission, but crucially also with regard to immune system. And what I think is also very interesting, what we can see on the right-hand side here is that, of course, most of the gene expression is relevant with regard to different brain regions, for example, the frontal lobe, but also other areas of the body play a role here, for example, the pancreas. And this is interesting because we also have a relevant overlap between schizophrenia and certain metabolic diseases, for example, diabetes, which also is a relevant driver of the increased mortality in these patients and the reduced life expectancy. And so on this slide, we can see some examples with regard to the relevant gene alterations, including a subunit of the NMDA receptor and the AMPA receptor, and then these proteins that are part of the cytoskeleton and that are relevant for long-term plasticity, long-term potentiation. Of course, genes are important, but as we have learned for so many diseases now, it's often not the genes alone, it's nature and nurture. So it's also often the gene environment interaction that we can observe as relevant causal risk factors for the disease development. So we have these existing kind of basic individual risk determined by the genes, and then we have the environmental factors, including the pre- and perinatal factors, the societal, the immunologic factors, also nutrition plays a relevant role here. And then we know that there are also brain morphological changes associated with schizophrenia. We can observe enlarged ventricles. We can observe alterations with regard to the interneurons with decreased GABAergic signaling. Interestingly, we find increased pyramidal cell density. We find decreased oligodendrocytes. They are usually responsible for the myelination, which, as we know, is decreased here as well. We find decrease of anatomical asymmetry. And so all these are characteristic morphological brain changes in schizophrenia. And of course, one interesting research question is whether these factors are causal for the development of schizophrenia, whether they are an epiphenomenon of the disease, or whether they affect each other both ways. And now I want to move on to what I think is also a fascinating research field, the neuroimmunological mechanisms of psychosis. And as we know, we have these different symptom categories, including the positive symptoms, the negative symptoms, and the cognitive symptoms. And we also know that perceived stress, including biological, physical, and psychological stressors, leads to an increased risk of developing psychosis, of transitioning to psychosis. And now the question is, okay, how does this emotional state of increased perceived stress now translate into these symptoms? So what is, for example, some kind of molecule that transfers this information of the organism being in stress to the brain, leading to changes in the brain that then cause these different symptoms? And one leading hypothesis here is that this is due to inflammation. As we know that chronic stress leads to chronically increased levels of cortisol, and this can then lead to a long-term low-level inflammation. And this increased pro-inflammatory status observed in the blood has also been shown on the meta-analysis level as being associated with a significantly increased risk of developing psychosis. And then on the other hand side, we also know that there are specific alterations, specific changes in neurosignatures, not only being associated with schizophrenia in general, but specific types of neural changes being associated with specific symptomatic outcomes. For example, the different neurotransmitter imbalances, but also changes with regard to the functional network architecture of the brain. So we can differentiate between changes more towards a more integrated brain network versus a more segregated brain network. And these are then associated rather with positive symptoms such as delusions, or rather with negative symptoms, including apathy, which I think is really fascinating. And we have also a number of studies showing that immunomodulatory drugs, so pharmacological interventions that fine-tune the immune system and often dampen it in general, can show positive effects, significant improvements with regard to the symptoms, especially with regard to cognitive symptoms. So these are really interesting and promising results, but we also have to mention that while these results are often significant, they are not yet clinically relevant, and this is not part of standard clinical practice to administer these drugs. One of the reasons being that the effect is very heterogeneous throughout the whole patient population, and so it is a relevant scientific challenge to identify a subgroup of patients that stand to benefit the most from these immunomodulatory treatment approaches. And so what are then the concrete underlying molecular mechanisms with regard to neuroimmunity in psychosis? So as I mentioned, we have these long-term low-level inflammation as an underlying risk factor, and this leads to an increase of these pro-inflammatory cytokines, so these information transferring molecules of the immune system. And these can then also traverse the blood-brain barrier in the state of inflammation, and then they activate the immune cells of the central nervous system, the so-called microglia, and they then trigger multiple mechanisms, activating T cells and then also B cells producing antibodies, but also being directly active through the generation, for example, of oxidative stress, of reactive oxygen species, which can then directly affect both the morphology and the neurotransmission in the brain. And crucially, what I think is also highly interesting, these pro-inflammatory cytokines not only activate the immune cells in the brain, the microglia, but they also have a direct effect on the neurons. So evolutionarily, the neurons have developed in such a way that they themselves also have receptors for those cytokines, including interleukin-6. And so their transmission activity, their activation thresholds, and many of their important characteristics can also be influenced directly through these interleukins. So it makes sense that some of the anti-inflammatory drugs show promising results in schizophrenia, while they are not part of clinical routine yet. And to dive into even more detail at the synapse, we can look at both the early stage and the late stage of the inflammation at the synapse. So at the early stage, we have this increase in pro-inflammatory cytokines, including TNF alpha, interleukin 1, and so on. And this then leads to an increase of glutamatergic release. And actually, the increase is to such a degree that all the released glutamate is not being defined to the synaptic gap anymore, but a so-called synaptic spillover occurs. So in the early phase, we really have a lot of glutamatergic transmission, a lot of vesicles are set free, a lot of glutamate is being detected by the receptors in the post-synapse, and then crucially, we also have a lot of reactive oxygen species, a lot of oxidative stress. But then on the long term, this leads to something that is called excitotoxicity, because not only the synaptic glutamate receptors are being activated, but due to this glutamate spillover, also the extrasynaptic receptors are being activated, giving the signal to the neuron that there is too much glutamatergic transmission going on. And so on the pre-synapse, less glutamate is being released, and on the post-synapse, less glutamate receptors will be present in the long term. So the glutamatergic transmission actually decreases in the late stage, and also due to this oxidative stress, we can have an increase in this so-called excitotoxicity. And this is also what we can see here in more detail. This especially is relevant with regard to the post-synapse, all this oxidative stress and the neurotoxicity this causes. And so to summarize, we have improved our understanding about the association between different kind of symptoms that can be grouped together in syndromes and which can then be caused by different disease entities. For this, we have talked about the autoimmune encephalitis, the NMDA receptor encephalitis as one important example here, and also schizophrenia, the so-called idiopathic psychosis, which is a clinical diagnosis. For which many different risk factors are relevant, are affecting the disease trajectory at both an early developmental, prodromal, and then full disease state. And many different mechanisms are relevant here, including the dopaminergic, the glutamatergic, and the GABAergic transmission, and changes in the function network architecture, and crucially also the neuroinflammation. And with that, it's my pleasure to hand over to you, Konstantin.
Thanks, Christoph. Maybe we can do a just 5-minute break to get some coffee or water, and then we'll see each other at, well, let's say 8 minutes, 17:30. Maybe you can hand over or can I just share the screen? Okay. All right.
I think
See you
that
at
should
this.
work, yeah.
Okay. Can you see my screen and can you hear me? Okay, perfect. So thanks, Christoph, for this expert introduction into the field. And I want to now show you some work of collaborators that Christoph also mentioned already from Munich, but also some of our own work that we did in computational modeling of psychosis and to better understand the disease and especially find possible ways to intervene in this so-called window of opportunity that Christoph mentioned. And so we already saw that psychosis is a very complex disease and it's not easy to find biomarkers. It's not easy to put your finger down what actually causes it. And it's definitely multimodal. And this can also be seen by this study by Friedman et al., where they showed that the inter-rater variability between two clinical raters varies greatly, which can be seen by this kappa factor. And this shows that there is a need for computational models that can better describe this disease. And there are different ways to approach this. So the very theory-based bottom-up approach is to find a hypothesis, to test it, and then to explain the disease bottom-up. Another one that is something that Nikos Kotsouleras did from Munich is the data-driven approach. So the top-down approach, meaning you have a lot of data and you try to find a find a data-driven explanation for this data that you see. And the third one, which we combined with the data-driven approach from Nikos, is a knowledge-driven approach using our tools, or especially our tool TVBase with other knowledge graphs and knowledge models. And the idea is to find biomarkers for the transition to psychosis or the transition to schizophrenia that not only show the clinical picture of the disease, but also gives us some quantitative measurement. And in psychiatry, there's one thing that's kind of like the best biomarker you can have is the so-called endophenotype. And that's a combination and from different criteria. And one is that it has to obviously segregate between illness and non-illness in the population, but also must be heritable and also takes into account the environmental genetic factors and must be non-state dependent, meaning that an endophenotype can not only be positive if you're in an acute psychosis, but must also be measurable in a down phase or in a non-psychotic phase, if you have schizophrenia, for example. And it must co-segregate between illness in families and must be present at a higher rate within affected families, obviously. And as I said, one top-down data-driven approach was from Nikos Kotsouleras et al., who did a large multi-center, multi-site study in Europe, where they overall measured 668 subjects, of whom 334 had either were at clinical high risk for developing psychosis, meaning that they already showed attenuated psychosis symptoms, but not actually had a psychosis already. And also the other half with a recent onset depression or some sort of as a proxy for the negative symptoms that Christoph described are becoming more and more relevant in the disease etiology and understanding. And what they did is they had different models, one including a SMRI at the beginning and after 9 months of the study, then also a polygenetic risk score and clinical neurocognitive assessments during multiple months, and then a follow-up period from 9 to 36 months. And what they were interested in is which of those people or which of those patients with a high risk of developing psychosis actually did transition to psychosis. And they used the three models separately, but also tried to find the optimal clinical scalable workflow, meaning that you only used as many models as you needed to find this transition phase or to predict the transition phase. And sorry, there you go. What was shown is that only a very small proportion of the patient actually transitioned to psychosis. So only 26 of those over 300 patients, and only a very small portion of those 26 actually developed schizophrenia, which is again just a demonstration that not all psychosis is related to schizophrenia, and there are different causes and different etiologies that can be attributed to psychosis. And what they did here, I don't want to go into all the detail, but they had a complex machine learning algorithm with a multi-step approach where they first tried to find the optimal sequential prognostic workflow. So which model should come first in order to have the best probability to find those patients with a transition to psychosis without using too much resources. And then in the second part, finding possible co-founders and optimizing the models. This also was done in a cross-validation loop with this multi-site study. So for each site, there's one model. And what we were particularly interested in as we're neuroscientists was the structural MRI model. And this consisted of two metrics, especially for the visualization. The first one was the so-called cross-validation ratio. And this describes higher or lower gray matter volume. So as you can see here, so red is a decrease and blue is an increase in gray matter volume. Gray matter volume in patients that transitioned to psychosis in comparison to those that did not. And the second metric was a so-called sign-based consistency, which basically said which of those voxels were consistently significant over all cross-validation runs. And there was a pattern of volume gains and also volume losses. So again, red is volume loss and blue is volume gain. And some of these areas were quite easily maybe explainable. So for example, volume loss in the superior temporal gyrus, which could be attributed to acoustic hallucinations, or volume gain in the dorsolateral prefrontal area, which could be attributed to thought disorders. But it is not that easy to explain why these volume gains and volume losses occur, and also in such a short period. So only in this transition phase, not as we saw before in Christoph's talk, in the full-on chronic schizophrenia disease. In the full-blown schizophrenia. And what we can see here is that the different models kind of performed equally, and if you stack those models, you had an even higher accuracy in predicting this transition. But as I said, we focused on this SMRI pattern that differentiated between the transition to psychosis and non-transition. And yes, so can we use this now? So how is this helping us in understanding the disease, and how is this helping us in clinically helping those patients, and especially what is the meaning of this predictive signature? So the SMRI signature, these volume gains and volume losses. And is there a possibility to find the mechanism behind the whole thing? And this is where we came into play, and we used our software TV base. I think you just saw it, so I'm going to keep the recap very short. Basically, this is a semantic mapping tool where we use the established knowledge base SkyView, which has a annotated PubMed literature corpus where you can search for annotated keywords in different ontologies. For example, the Uberon ontologies, so meaning you can search for anatomical terms. but you can also search as in a Google search for free text, but also for things like the gene ontology or for gene names. And what we did is create a so-called transformation matrix where we translated these Uberon terms into a 3D parcellation, 3D template brain, where we used the Glasser parcellation for cortical areas and the Fischl areas for the Fischl parcellation for segmentation for the subcortical areas. And what this allowed us is then to search for certain queries, for example, a gene name or a protein name in the context of psychosis. And this gave us a semantic map which showed which areas of the brain are most relevant for your search query, if there are any. And we then validated this kind of new form of data with different empirical data, especially with this paper from Marcello et al., where we showed that these two-base maps, if you just search for the semantic query of alpha or beta or delta theta rhythm, this correlated pretty well with the actual empirical MEG rhythms that you saw spatially, also for PET tracers and also for other measures for brain organization like cerebral blood flow, glucose metabolism. So this was, so to say, our data form that we used. And this is now a overview about the whole methodological framework. And what we did is we used HCNC, so a ontology for all known gene names in our genome, and created semantic maps for all of these genes, and used these then as a input feature to decode the transition pattern in the SMRI from Cortularis et al., which was created by the cross-validation machine learning algorithm that I showed before. And the second step, we then try to interpret our model results. So what this actually gave us is a kind of ranking or feature importance of each map. And this was then further interpreted with using string, which I'll go into in more detail later on. Yeah. So basically 3 parts. We created the maps using TV-base and HCNC. We then used a support vector regression to decode this cross-validation ratio map from Cortularis et al. Then used a string to actually explain or better understand what we found in this support vector regression. And one thing I have to mention here is that we actually did kind of 2 different mappings. So one's without context, meaning that we just searched for the gene name and saw what relevancy came out disease agnostic, and also one's for the psychosis query. And basically this pathway database circle kind of represents everything that we know so far about disease-specific pathways, but this doesn't mean that we actually anywhere close to the true psychosis pathway. So we are limited with TV-base to the current knowledge that is described in the public database. So we are aware that we are not sure how much overlap we actually have about the true knowledge about psychosis pathways. And another thing that we did for our support vector regression is that we binarized relevancy areas. So meaning, as I said, the semantic association maps give you a relevancy value according to the literature. And what we wanted to do is to try to reduce the publication bias, because some areas, especially like you can see here, like hippocampus, are researched more often and more detail and tend to be over described, and so lead to a higher relevancy. And if we binarize this relevancy, you kind of can reduce this or remove the publication bias. And also we wanted to give the support vector regression, more emphasis in finding our in finding the association to the actual transition map from Cordilleras. And the results of this support vector regression were quite strong. So if we used the no context maps, meaning agnostic to the disease, we actually had an R squared from at around 0.65. And if we use the disease-specific context in psychosis, so we then looked for the gene name or the protein name, and in the context of psychosis, we had a R squared from around 0.3. These blue box plots you can see here are actually a null model where we shuffled the target maps of the cross-validation ratio map 100 times and saw how specific the our feature set is to the cross-validation ratio that we saw, or if it's just good at kind of decoding any possible map. And what we see here is the class delta, meaning a effect strength. We can see that both models kind of performed the same, but the more specific psychosis model did perform a little bit better. And yeah, I just have to mention this one thing we did with the cross-validation ratio map. So the final distribution CVR map that we see in the slides before is that we mask the cross-validation with the sign-based consistency. So to only look at voxels that were consistently significant over all cross-validation runs. And then in the end, so this was voxel-based, and then in the end also transformed this in our two-base space consisting of the cluster parcellation and the subcortical facial areas. And this is then how the reconstructed map looked if we just kind of aggregated the SVAR feature importance and the TV base maps. And as you can see here for the no context, this is very much the same with the most relevant areas, or the most volume gain and volume loss areas quite accurately depicted. And the whole idea was obviously that if a gene or a protein is relevant in a specific area, it's also likely that it will enact some function in that area, right? So if an area, if the genus acts mostly in, or we know that it's mostly, sorry, one second, I just have to open the door. Sorry for that. If we know that a gene is mostly expressed in a temporal lobe, it will mostly also enact its function in this area. One thing we were mostly interested in is not particularly to just kind of reconstruct with the highest accuracy, so just reconstruct the pattern to a perfect extent, because we had one problem that we just looked at one target map with a large feature set. So we had a very strong discrepancy between this large feature set and the target map. And we actually tried to use this support vector regression to give us an idea or use it as a feature selection, so to say. So not just use it as a reconstructive pattern, but also show which genes are most strongly related, or which semantic association maps from genes are most strongly related to this pattern and most consistently. And this is what is shown here. So we not only used the semantic association maps from TB-based, but we also used gene expression data from the JUGEX data set. And what you can see here, that this is also great in reconstructing this pattern, but it's not very specific. So there's only a very small significance between the reconstruction of the transition pattern and the null model. And what we also can see here in these two plots and here, as I said, the reconstruction with the no context is better than the psychosis context. But here, if we increase the threshold value for the relevancy areas in the semantic association maps, we can see here that for the psychosis query, for the psychosis context, we have the slowest drop in the model performance. So likely the good performance of the no context map model is also based on the density of very low value, low relevancy values in a lot of areas, and just having a larger feature input, right? So because with the inclusion of the psychosis context, we have less knowledge specific to each gene. This is why we then for the further analysis focused on the psychosis context model, and what we're interested in is to find the most relevancy, the maximum relevancy and minimum redundancy features. So those features that are always used for the reconstruction. That's too specific, maybe. But basically, what we could find in a further analysis is that if we use a forward feature selection algorithm, so we kind of put one more map into the model and see if this increases the model performance or not, and if not, then we deselect the feature, and if yes, we select it into the model. We see that the peak is reached around 40 feature maps, and that some of these feature maps are kind of always selected, independent of which model we are or which gene we're starting with. Some feature maps are always used, and this is then the final metric we use for interpreting the significance of each map for our model. And this is the result of this support vector regression. And now on to the last part of this work is the interpretation of these maps and of these feature importance maps. As I said, we are kind of expecting that if the gene is active in one area, it will also enact some function in theirs. And that's why we use the string database to have some interpretation of these acting genes. And the string database is a very large knowledge base containing the protein-protein interactions that are known so far. It's constantly updated. I think the last update was 2023. And what this allows is to actually have, so with these features that I showed you, to have a protein-protein interaction network, and then interpret firstly if this network is significant in a way that the interactions that we see in this network are more likely than in a random network of the same size, and also gives us the opportunity to interpret the interaction that these proteins would likely have on a molecular scale, right? And what's also possible with the string database is to segregate your data, your protein-protein interaction network into different clusters, each then having also a different function or a different functionality. And what we use to differentiate these clusters is the so-called Markov clustering algorithm, because this was shown that it's quite robust for the protein-protein interaction networks. This is based on a random walker, and you have the possibility to increase the effectiveness of very close relationships with the increase of the so-called inflation parameter. So with a higher inflation parameter, you get more segregation in your network. And when we applied this for our 40 features, which actually came down to 46 genes, because some of the feature maps were, because of the binarization, were kind of equal. So we subsumed these into one feature. So some feature maps just basically looked exactly the same. So we substituted these into one feature. This is why we came up to 76 gene maps. And if we look at the biological interpretation of these maps with an inflation parameter of 1.4, the strongest performing cluster, or the strongest performing genes, protein-protein interaction network within our cluster was one that is related to interleukins, especially with interleukin 6 here as a hub node, inflammatory response and neural growth. And how interpretation of this is done also using string, where you can look at the functional enrichment, meaning that you have a look at which biological processes, for example, in the gene ontology, these genes are associated with, and then test how likely is it that you have this many genes associated with that biologic process within a set of this genes, set of this size. And the second biggest cluster was one associated to neurotransmitters, especially dopaminergic and glutamatergic signaling, which is kind of nice because we know that this is also a very basic pathophysiological feature, as we saw before in Christoph's talk. And the third cluster was also associated to neurotransmitters and GABAergic signaling. And we did this for different inflation parameters with higher and higher segregation. And we came down to this area of significant functional enrichment between the inflation parameters of 1.4 and 1.7, meaning that increasing these inflation parameters didn't lead to any more significant clusters, and decreasing this inflation parameter did not just left us with one giant blob, so to say. And we can see here that the segregation then comes down to different neurotransmitter systems and also neural growth and inflammation, the cluster I showed you before. And this was quite consistent with different inflation parameters. So what does this tell us? We can see that we can decode this transition signature that we saw from the data-driven approach from Nikos Kotsouleros et al. We can decode this using our semantic association maps, and not only decode it, but also find meaning behind it. Yeah. So when we think back, what the maps actually meant was a decrease and increase in different gray matter volume parts of the brain. So it's very conceivable that, or makes sense that inflammatory processes, for example, lead to a volumetric increase just by edema or other factors, and neural growth, plasticity or reforming of the brain during the disease or because of the disease also makes sense that this leads to volumetric increases or decreases. And it is also, as I said, nice to see that most of the transmitter systems that we know that play a part in the pathophysiology of psychosis also came up as possible explanatory processes. And one recent work with Kotsouleros et al. Also used the same Pronia cohort to look at different targets or further targets, and another target was the so-called the remission. So it's not exactly the same patients, but it's the same cohort, and they had a look at a signature also again in this gray matter volume that was predictive of the remission of those patients. And this was defined according to the remission in the schizophrenia working group. Basically meaning that you had to have a significant decrease in the psychotic symptoms and also a temporal stability criterion. And this pattern we again decoded, kind of saw the same pattern that the no context model had a higher reconstructive power, but was less specific in this part, even more so. And the interpretation of these patterns again showed some known processes, some known explanatory features. but also some new ones, especially if you look at the yellow part, we still have the inflammatory pathway, the inflammatory cluster. Here we also have the neurotransmitter cluster, but if we have a closer look at what's different between those two models, so here shown as a waterfall plots, meaning that these genes over here perform better in the remission dataset and this performs worse in the remission dataset. We can see that overall, better for the remission of people or of patients who transition to psychosis were genes associated to neuroplasticity. So meaning if you have well, the interpretation is kind of abstract. But possibly it's easier to find remission if you transition to psychosis if you have a more plastic brain that's able to adapt, and at the same time it's genes performed worse for the remission decodation, which were associated with behavioral or cognitive genes. And if we use a different ontology or different knowledge base for the enrichment analyses, so now in the human phenotype ontology, we see that genes that are associated with negative social behavior perform better in the remission model, and those that were related to neurodegeneration, lipid metabolism, performed worse. Yeah, which was quite interesting. But again, this is very, very new work. We have to test this. Also, for the remission model, we did not have cross-validation data, so this is kind of a very early comparison, so to say. Okay, so as I said, the main pathways now, coming back to the transition model, are the inflammatory response, neural growth, and neurotransmitter imbalances. And if we think back, why are we doing this? What is the whole purpose of this decodation model? We want to find mechanisms that we can target clinically and pharmaceutically in this window of opportunity. So we're looking at people who are at the verge of transitioning and that are then actually transitioning. So what are the mechanisms that can lead or that can explain the transition to psychosis? And if we think back to our clusters, we actually have some possible targets. For example, in the inflammatory interleukin cluster, we would have interleukin 6, which can be inhibited with tocilizumab, or which is an antibody especially working against interleukin 6, and which was also shown to reduce the EL6 caused excitation in this mouse brains. So with this knowledge-driven approach, we possibly have a agnostic tool that can lead to a hypothesis what we could then actually use clinically or pharmaceutically to reduce the transition to psychosis in patients with a high risk to develop this psychosis. And this is a closing slide that I like to show in the end, because this is kind of it's called ontologic loop that I just mentioned. So we're using actual empirical data, the CV ratio data from Consuleros et al. And then just by using these knowledge bases and knowledge tools like TVBase, we can find possible mechanisms of possible testable hypotheses that we can then infer into the virtual brain for in silico testing, or that can be used for animal testing or anything. And especially with if you remember the talk from Leon Martin, this also fully automated with the TVB ontology. So we kind of have a pathway or a loop that we can use without any real human interaction or any hypothesis generation that we need to do a priori. So we have this empirical data, run it through this loop, and also simulate possible mechanisms in TVB. which is something that we are working on right now, but we cannot show you already. And this is just an explanation of possible candidates that could be tested in silico within TB-base, so especially the altered excitation inhibition balance for inhibition of GABAergic interneurons, but also for inflammation, because we know that IL-6 can lead to more excitation in particular brain areas, but also, for example, a multi-scale model between the D2, D1 receptor densities. Yeah, and then I want to hand over again to Christoph, who can show us some more work that's being done on validation one of these hypotheses, especially the inflammation hypothesis, on an external data set. Should we switch again, or should I show the slides?
I think it's great if you can just click through the slides. Thank you very much,
Yep.
Konstantin, for this wonderful part of our shared presentation. Highly interesting work and so much more to come. Exciting to be a part of this. And as you mentioned, exactly, we are now also moving on to validate this candidate mechanism of the neuroinflammatory processes with an external data set. And for this, we choose the Human Connectome Project for Early Psychosis dataset, which is a really high quality dataset collected in the US, among other centers also in Boston. And this is a dataset with over 250 subjects. And we have randomly selected a sample for a pilot study that we have conducted with 10 subjects with an average age of 24 years and 50-50 sex distribution. So this is a pilot study, a proof of concept. And for this, we used different metrics of symptom severity, especially the positive and negative symptom scale, which is also widely available in clinics. Then we use different modalities of brain imaging, specifically with regard to structural and functional MRI. And then we wanted to also include markers of inflammation. And here it's important to point out that this is very preliminary work, proof of concept, and thus we have used imaging-based inflammatory biomarkers based on the literature. We chose total cortical thickness and total brain volume, which have been shown to be associated with also immunological markers in the blood with pro-inflammatory cytokines. And of course, this can only be the first step. We are looking forward to extend the whole study and then use also advanced imaging-based inflammatory biomarkers, including free water imaging and also biofluid-based inflammatory biomarkers, for example, taken from the blood. And then we can see on the next slide that the basis of the analysis, of course, is that we want to replicate the association frequently reported in the literature, that we can find an association, a statistical association between the levels of inflammation and the symptomatic outcomes, specifically with regard to the cognitive symptoms. And this is the basis for the mediation analysis we then want to perform, evaluating how much of the effect of the exposure variable of inflammation on the outcome variable of the cognitive symptom severity is being mediated by changes in the functional connectivity, which we assessed using the functional MRI imaging, creating this functional connectivity matrix, and then analyzing this as a first step with two very general parameters of global connectivity, one being the marker of integration, which is global efficiency on the one hand side, and then a marker of segregation, which is the clustering coefficient on the other hand side, which have been established in the literature already. And we were able to replicate the findings from the literature with regard to the effect of inflammation on the symptomatic outcomes, showing a strong statistical association and a strong correlation of around minus 0.65 or minus 0.64 respectively for total brain volume and total cortical thickness. And in this very small pilot study sample, the p-values already are on the edge of significance, and we expect this effect to only become stronger in the full sample, which we have already started to analyze. And so as this basis has already been established, we then wanted to know how much of the variability in functional connectivity patterns can be explained by the total brain volume and the total cortical thickness. And together they explain around 83% of the variability here, leading to a p-value of 0.023 adjusted for age. So we have a very strong and also significant association here. And then moving along, we also wanted to evaluate the association then between the functional connectivity metrics and the symptom severity. And this analysis showed that around 61% of the variability in symptom severity can be explained through changes in total brain volume, total cortical thickness, and the functional connectivity marker of global efficiency adjusted for age. And taken together, this now enables us to evaluate the proportion mediated. So how much of the effect of inflammation on the symptomatic outcomes is being statistically mediated through changes in the functional brain network architecture. And I want to emphasize that we really used as a proof of concept two very general markers of this network architecture. The first one being global efficiency, and the data actually shows that over 19% of the effect of inflammation on the symptomatic outcomes is being mediated through changes in global efficiency, and another 8% is being mediated by changes in the clustering coefficient. So taking together these two very coarse, very general markers, we can see that around a quarter to a third of the effects are being mediated just by these two markers alone, showing that a relevant proportion of the effect of inflammation on cognitive symptoms is being mediated by global parameters of functional outcome. And we're having ongoing work expanding this study now also to our full data set, also including advanced markers of inflammation in this process. And as we speak right now on our clusters here in Berlin, we also have the ongoing brain simulations and then linked to these functional connectivity patterns to evaluate also further mechanisms with regard to the underlying cognitive processes. And with that, we of course want to show our multiple references that we have used for this talk. And I think crucially, we also want not only to thank you for your attention, but also want to thank our PI, Professor Petra Ritter, and all of our wonderful colleagues, without whom all of this great work would not have been possible. Also our collaborators, of course. And now we're looking forward to your questions.